Я ще раз сердечно вітаю всіх на Буковині в нашому навчальному закладі.
Це надзвичайна подія.
BIMCO is a life-changing experience.
Stace is here somewhere, so she's called me about 200 times.
Робота кожної секції включатиме наукову дискусію з обговоренням критеріїв і визначенням переможців.
Я ще раз сердечно вітаю всіх на Буковині в нашому навчальному закладі.
But there's no one who's falling down each step.
Much better, and then you can write it down, and you can ask me later if you want. I'll write it down for you. Different sizes. Second shot, CPR. Third shot, adrenaline, amyloid. Okay, cardiac arrest. Call, you put the cardiac arrest key down. Okay, stop. Everyone say oxygen away. Noise is now. And flush it. Different sizes. Be careful when you use this, because if you have
Good morning, dear participants of Bukovinian International Medical and Pharmaceutical Congress of Students and Young Scientists. Welcome to BIMCO 2020. We have just seen the brightest and the most memorable moments of BIMCO 2019. These unforgettable memories warms our hearts today when the usual and planned way of holding BIMCO is impossible. But we are very proud and happy that no obstacles can prevent BIMCO 2020. And this year, Bukovinian International Medical Congress is taking place online. Every year, our international congress unites young scientists from different parts of the world and make their hearts beat together in the rhythm of love for science, for medicine and for BIMCO. And this year we sincerely congratulate members of educational institutions of Ukraine, Australia, Albania, Brazil, Bulgaria, France, Germany, Ireland, Italy and many other countries from all continents. All members of BIMCO organizing committee, all professors, teaching staff and students of Bukovinian State Medical University express the gratitude to every member of international BIMCO family. Today, our university has opened its doors for all of you. Therefore, we can say that Bukovinian State Medical University is known not only in Ukraine, but beyond its limits, and we have a lot of things to be proud of. And now, with great pleasure and gratitude, the floor is given to respected and esteemed director, the Chairman of Organizing Committee of BIMCO, Professor Taras Mikolayevich Boychuk.
Доброго дня, вельмишановні учасники Міжнародного медико-фармацевтичного конгресу. Цьогоріч, враховуючи особливу ситуацію у світі, пов'язану з пандемією коронавірусу, ми проводимо наш конгрес в онлайн-режимі. Тому висловлюю подяку всім, хто підключився до нашої трансляції. Насамперед, висловлюю особливу шану і подяку медичним працівникам всіх країн, які сьогодні борються за життя людей, демонструючи людству неймовірну відвагу і високий професіоналізм, інколи навіть ціною власного життя. Колись вони також були студентами, а сьогодні їхні вчителі пишаються відданістю своїх вихованців. Такі ситуації підкреслюють виняткову значимість медичної професії, важливість високої кваліфікації лікарів, медичних сестер. Переконаний, що наш медичний конгрес є ще одним кроком вперед до вашого професійного зростання. Я бажаю всім міцного здоров'я, цікавих дискусій, знайти нових друзів, з якими ви обов'язково зустрінетеся за більш безпечних умов. Нехай вам щастить!
Thank you, dear and respected Rector, Professor Taras Mykolaievich Boychuk. And now I would like to invite the leader of Student Scientific Society, the head of the Council of Young Scientists, the associate professor of the Department of Pathological Anatomy of the Bukovinian State Medical University and the co-chairman of BIMCO Organizing Committee, Тюленєва Олена Анатоліївна.
Вітаю учасників Міжнародного медико-фармацевтичного конгресу BIMCO, який традиційно у квітні проходить на базі Буковинського державного медичного університету. Традиційний конгрес став місцем зустрічі молодих людей, перспективних науковців, однодумців, чиї інноваційні ідеї є надзвичайно цінними для розвитку медичної і фармацевтичної галузей. Цю подію щорічно з нетерпінням очікують не тільки учасники з різноманітних куточків світу, але й члени організаційного комітету. Кілька днів конгресу дарують море позитивних вражень, нових ідей, як для учасників, так і для організаційного комітету. Багато хто з вас став справжніми друзями BIMCO, беручи участь у конгресі не один рік поспіль, стають нашими амбасадорами. Сьогодні науковий форум проходить вперше в дистанційному режимі, і 400 його активних учасників з 17 країн, я впевнена, отримають не менше вражень, позитивних емоцій, нових ідей, натхнення, ніж від традиційного формату проведення конгресу. Організаційний комітет BIMCO зробив усе можливе для того, щоб кожен з вас насолодився плідною науковою співпрацею в комфортних умовах онлайн. Тож я вам щиро бажаю натхнення, успіхів, невичерпної енергії, оптимізму та віри у свої знання. Удачі!
Thank you, dear Elena Anatolievna, for your speech. And now we are having an opportunity to listen to the words of greetings from the Congress participants and guests who is representing our BIMCO media partners. First of all, I would like to invite Dr. Ryan Kalaso, emergency medicine physician from India, medical editor at USMLE IFOM, manager for the Berlin headquarters of AMBUS. You're welcome.
Hello, everyone. My name is Dr. Ryan Kalaso, as your moderator just told you, and I welcome all of you to BIMCO. I hope that you all are keeping safe. It is quite the unique time that we all live in, and it's the first time in our lives that everyone around the world is going through something together. And I hope that in this time you guys are keeping safe and well informed. And for those of you who are in practice, I really hope that your hospitals have adequate PPE and that you're staying on top of the emerging literature. It's an ever-changing situation. We know how it is. Well, first of all, congratulations to the BIMCO Organizing Committee and to all you participants for adapting so well to this changing reality. For a conference to go completely online and to do so smoothly is just amazing. And congratulations to you. We are really happy to be associated. AMBOSS is really happy to be associated with BIMCO. To all of you, what I'd like to say is, I'd like to inform you, I'd like to welcome you to the conference and inform you that myself and Dr. Taner Shrank, who is the partnership manager for Eastern Europe in AMBOSS, we both will be available at the conference. We'll be holding a little workshop tomorrow to show you Amboss and to show you how Amboss has helped so many students around the world do better at their medical studies and become better doctors. Of course, for those of you who are studying for the USMLE or for the IFORM exams, you may have already heard about Amboss, may have even used it, and know that it is this leading international study tool. To the rest of you, what we'd like to do is show you how effective studying medicine can be, and yeah, in general, just show you we are a team of about 60 doctors from all over the world who've come together to make medical studies easier for you, to not have the same problems that we faced when we were students. So I hope that this works, that you all can come to the workshop. And what the two last things that I'd like to say is that everyone who attends the workshop, the Amboss workshop tomorrow, will get 15 days of free access to all of Amboss's questions and learning cards, which is chapters, for 15 days to try it out for free. This is complimentary to everyone who attends the workshop. Additionally, all the prize winners in at the didactic and academic events of BIMCO will be winning 3 and 6 month accesses to all of Amboss. So watch out for that, and really hope I really hope that you participate well and that you enjoy this conference. If you have any questions whatsoever about Amboss, please reach out to your organizing committee and they will put you in touch with us. Thank you, welcome, and stay safe in these times. We are really excited to see you tomorrow at 11 o'clock at the Amboss workshop. Take care.
Thank you, dear Mr. Colasso. I'm sorry, 10 a.m. according to Ukrainian time, GST +3. And thank you once more time. And I would like to invite the next speaker, the representative of Lecturia, partner of BIMCO, Kwetsimani Shipalana, medical student at Vinnytsia National Medical University, the ambassador of Lecturia from South Africa.
Hello, everyone. I hope you can hear me. Thank you to the organizers for this opportunity. I am Kwetsi, a medical doctor currently studying at Vinnytsia National Medical University, and I am a Lecturia ambassador. So I'm just going to talk a little about what Lecturia is about and my experience with Lecturia as I have used it before. So Lecturia is a platform which provides detailed topics and detailed stuff about medical stuff for medical students, obviously. And I have personally used it. It has videos, it's got question banks. And my personal favorite thing about the question banks is that it's not only used to test yourself, but it can also be used for knowledge. Actually, when you get an answer wrong, or all the answers have detailed explanations as to why this could be the answer or why this shouldn't be the answer. And so Lecturia, being a partner of BIMCO, we are offering a 3-month premium membership for the winners of the conference, of which Irina would be able to contact you with regards to that. And for everyone participating in the conference, we are giving a 50% discount for the next 2 weeks should you sign up for Lecturia. You will be getting a percent discount for all of our plans. So it can be the 1-month plan, 3-month plan, 6-month plan. We also have a 2-year and 4-year plan that we've recently introduced. Another thing is there have been updates on coronavirus itself, about how the statistics about treatment, about what is working so far, about who is more susceptible to coronavirus and all of that. So Lecturia is really just a great platform, not just for our studies in general, but about what's happening in the world right now with regards to the pandemic. If you have any questions, you can email your questions directly to support@lecturio.com, and we will get back to you as soon as possible. Thank you so much for this opportunity, and happy conference and happy learning.
Thank you, dear Kwetsimani, and thank you, Culturio, for having a partnership with Bukovinian International Medical Congress. And now I would like to give the floor to the next speaker, Christos Stakaris, a medical student from Greece, who is our ambassador, who is also the Novel Med editor-in-chief and the core facilitator of the Students Against COVID movement.
Thank you. Honorable professors, dear friends and colleagues. It is a great pleasure for me to be here today. I have the privilege to be a member of a BIMCO family for 3 years. As a student ambassador for my country, Greece, and my university, as editor-in-chief of NovelMeds, a student journal where we try to publish some of the work that is distinguished here in BIMCO. 3 months ago, this speech and the whole ceremony would be different. However, the situation with the coronavirus is such that I now feel obliged to address you as one of the core facilitators of Students Against COVID movement. This movement, Students Against COVID, was started 2 weeks ago by Marina Kamal, a medicine and public health student from the University of Michigan. I was happy to join this movement from the very beginning, and since then, from a core team of 5 facilitators, we have now grown up to a team of more than 150 volunteers, students, and allies from more than 35 countries, stepping up against coronavirus during the pandemic. There are a lot of initiatives from students and young people in this situation all over the globe. What is different about this movement is the fact that it brings together as many as possible of them. We cannot, of course, take the credit nor the liability of each other's activities and initiatives against the coronavirus. However, we can all learn from each other, we can exchange good practices and build coalitions. And although all of us, most of us, are young in medicine and have limited experience with handling the pandemic, we have a lot of creativity. And this creativity can lead us to solutions that we may not imagine now. So, from this floor and from this point, I would like to invite all of you who want to join the movement using the hashtag and also joining the working groups we are now forming. This is all about the Students Against Coronavirus. Once more, I'm really happy to be here in the opening ceremony of BIMCO. It is an amazing platform where we can all learn from each other and interact in a meaningful way. And I wish all the success to the organizing committee of the conference. And I also wish that we all get the most out of it. Thank you very much.
Thank you, dear Christos Tsagaris, for your speech. And now the floor is given to the next speaker, Dr. Mashkur Isa, Youth Ambassador of European Code Against Cancer and member of Association of European Cancer Leagues, representing Ukraine, Nigeria, and United Kingdom. I'm sorry, Mashkur, can you please turn on your microphone? Mashkur, I'm really sorry, but we cannot hear you. We can see you well. Okay. I'm sorry. Unfortunately, we cannot hear Mashkur's speech, but I'm sure that he promised us all the best because he is not only representing European Code Against Cancer, but he also represents us as one of the coordinators of BIMCO Ambassador Program, being our ambassador and member of organizing committee for the last 3 years. Thank dear Mashkur for his speech, but unfortunately, due to some technical issues, we was not able and we are not able to hear him. And now, unfortunately, Mashkur do not hear me as well as I see, but I would like to invite the next speaker. And the next speaker is Eliana Shvachi, PhD student at Faculty of Medicine of Mashkur, I see you can hear us now. Unfortunately, you did not turn on your microphone. I will give you a possibility to talk later after Liana, if it is okay. Try to solve your technical issues. I'm sorry. Liana, please turn on your camera. Liana, please turn off your camera, Mashkur, for a few seconds. Yeah, I know. And Liana, please welcome. Liana is PhD student at Faculty of Medicine of University of Lisbon and University Medical Center of Göttingen. She is also our ambassador. Please welcome Liana.
Hello, good morning to all the present. My name is Liana Shvachi. I'm a PhD student in Biomedical Sciences from Faculty of Medicine of University of Lisbon in Portugal, and I'm currently working on my PhD project in University Medical Center Göttingen in Germany. I am very happy to be a participant and ambassador of BIMCO for the 3rd consecutive year. For me, as a Ukrainian by origin, it is such a great honor and pleasure to be in such a high standard conference in my home country. I would like to thank the whole organizing committee for their tremendous work and also for their creativity and ability to work under pressure as this year changed everything. And they managed to transform a huge conference in spot to an online congress in a very little amount of time. I would like to also personally thank Anastasia for her proactivity and great organizational skills. I'm really sorry that we could not meet in person due to the current situation, as BIMCO for me is like a meeting with friends, enjoying the time and also learning and sharing a very high standard science. I hope that all of you new and all participants will enjoy this meeting, acquire new knowledge and share your work in a most productive way. Welcome to BIMCO 2020 online version.
Thank you, our dearest ambassador, Eliana Shvachi. And now I would like to give the floor to Lorenza Bacco, PhD student at Paris Descartes University, Young Opportunities Organization representative from France. Lorenza, are you here? Unfortunately, the online format of the Congress changed something, and sometimes people just do not manage to connect in time due to some technical issues and some troubles with their connection. We are very thankful to all our guests and to all our participants and to all people who had a possibility to have a talk during the opening ceremony of our Congress. But getting new knowledge, sharing experience is actually the main task of our meeting. Famous scientists will teach us and share their experience. So now, with great pleasure, I would like to invite Dr. Tiago Fleming Otiero, full professor of agrigopathies, director of the Department of Neurodegeneration and Restorative Research from Göttingen, Germany, who is going to talk about From the Kitchen to the Bedside, Unraveling the Molecular Basis of Parkinson's Disease. I am giving the floor to our honorable speaker.
Hi, good morning, everyone. It's a great pleasure for me to be here with you using this different format for meetings that is proving to be very useful and allowing us to connect even at times when we cannot be physically together. So it's unfortunate, but that's life. So we cannot always predict what life brings us, and we have to adapt. And I congratulate the organizers, Anastasia, and all the colleagues that were involved in putting this meeting together and making sure that even despite this weird times that we're living in, that we can still have a nice scientific meeting. And so for me, it's a great honor to participate online. I would have liked very much to have come to Ukraine, a country I've never had a chance to visit before. So this is not going to be possible this time, but I hope that maybe next time I'll have a chance to come visit and participate. So now I will try to share my screen so I can show you my presentation. Can you see my presentation already?
Maybe we just have to wait for a few minutes.
Okay. I think you can see my presentation, right?
Yes, now it is uploading, and now we can see.
Okay, great. So I will put it in presentation mode. And okay, so I believe it's working now, right?
Of course.
Okay, good. Great. So then let me start. I hope you enjoy the presentation I have for you today. I came up with a bit of a strange title, From the Kitchen to the Bedside. What does this mean? Are we going to talk about food or something related to medicine? And I hope you will understand what I mean with this. So we work on different neurodegenerative diseases in my lab, in particular on Parkinson's disease, but we use Parkinson's disease just as a model disease. It's not that we are only interested in this particular neurodegenerative disease, but we think that it can be a good model to understand what happens in other diseases. And I could not start a meeting or a talk in a meeting these days without mentioning this agent that is changing our lives, as we discussed already before. So I thought maybe instead of talking about Parkinson's, we can talk about coronavirus, but I'm sure everyone is already sick of hearing about coronavirus and how it's impacting our lives. So in the end, I decided to just show this picture because it actually captures something that is related to what I will tell you about. And this has to do with what is inside the virus. And you will see here in this picture that it's cut, so there's this slice that allows you to see to the inside of the virus. And the point here is that even a virus, such a small particle that is a lot simpler than a cell, is very crowded internally. And you see the lipids here in green and these proteins in red that form the crown that gives the virus the name. And then inside, when you try to look inside, you have the nucleic acids, the RNA molecules, and other proteins that then allow the virus to cause the problems it causes to our cells and, more importantly, to our societies. So I like also to talk about things in simple terms. And there was a very famous scientist that you all know about that said very important things, like everything should be made as simple as possible, but not simpler. And then if you can't explain it simply, you don't understand it well enough. So sometimes to explain complicated ideas, you don't need to go and talk about all the little details and show that you know all the little details. It's sometimes harder, and it's often more useful to use simple ideas to explain complex problems. So with this in mind, I want to tell you what we do in my lab, what we study. So we study this process that is represented here in this picture. We study what happens to proteins, like the proteins inside the egg, that causes them to change conformation and to acquire different biochemical and biophysical properties. So what is happening here, what is represented here in this picture, is that because of the temperature that we use to fry the egg, the proteins that would normally exist in a normal state are changing their conformation and are becoming misfolded. And we know that this problem of protein misfolding is associated with almost all kinds of human diseases. It's associated with cancer, with diabetes, with neurodegeneration, and with many other types of problems, including also influenza or COVID-19. You can also think of situations where, because the cells are being infected by this virus, and the virus is hijacking the cellular quality control machinery, this is leading to the misfolding of other proteins. So this is, we think, a very central problem in biology. And in the context of neurodegenerative diseases, I'm sure you heard about most, if not all, of these diseases. We have Alzheimer's disease. This is by far the most common neurodegenerative disease, affecting millions of people around the world. It's followed by Parkinson's disease, which also affects a lot of people around the world. And then there are other diseases shown here that affect not as many people, but still have very devastating consequences for everyone's lives. And I'm sure at your age, you already met someone that has or had Alzheimer's disease or Parkinson's or one of these diseases. And this really shows how common these diseases are today. And the problem is that they will become a lot more common in the future. We'll come back to that. Another problem is that clinicians, so medical doctors, they can detect the tip of the iceberg. They are good at detecting the symptoms. And this creates the situation that the therapies we have today, when we have them, which is not true for all diseases, but they treat the symptoms, but they don't treat the causes. And this is what we need to study. We need to understand what are the underlying causes of neurodegeneration. And we think that by understanding the causes, we'll be able to develop novel therapeutics. So this is where we need to put a lot of energy still today. There's a lot of research going on in the world, but we need to do more to understand the causes so that we can come up with novel therapies. What we also know is that genetics explains only a minor fraction of these diseases. In some cases, genetics or environment don't really fully explain, as I'm sure you would agree. But this picture is just to show that it affects anyone. You can be a sports person, you can be an actor, you can be a president, you can be the Pope. It doesn't choose race or job or anything that we could use to stratify society. So it's transversal, it affects everyone. And we know that genetics, in all of these cases that are shown here, genetics doesn't seem to explain why these people developed Parkinson's disease. So there's other factors that explain what is going on. And we then face questions such as, why do some of us end up developing these diseases and others don't? And also, how can we diagnose these diseases so that we can treat them earlier? This is also a big challenge because these diseases are age-associated. They develop usually later in life. And how can we detect them earlier that we can start treating people before their symptoms are already too severe? So we know we are all different. I mean, this has been known for a long time. We are taller or shorter. Well, we have brown hair or black hair or blue eyes or green eyes. It doesn't matter. So we're all different. And this tells us that genetics, of course, is important, but also the way genetics interferes and talks to the environment is also extremely important in the context of these types of neurodegenerative diseases. And the other factor that I already mentioned is that our society is living longer. So we are, these are data for the UK, for men in blue and for women in pink. So you can see here in these curves that our population is getting older, we're living longer, and this means that there's going to be less younger people and more people reaching older ages, and this is creating a lot of challenges. Again, some of them we are witnessing today. People that have older ages, they are more vulnerable to viral infections like the one we're going through at the moment. So this is also relevant in the context of infectious diseases, but in the context of neurodegenerative disease, this is really extremely relevant, because we know that the majority of people that develop these diseases are older. So there's some aging component that we don't fully understand necessarily. So what happens? What happens is that if we go back to basic cell biology, all of you have studied cell biology in some way or another. And if you go back to any textbook, you find pictures that look like this, right? Everyone recognizes a cell, a eukaryotic cell here in this picture. You recognize the mitochondria, the nuclei, the ER, the Golgi, all the organelles, even these little dots that represent the ribosomes. And then there's this blue space here that tries to represent the cytosol. But the problem is that this is not a good way of representing the cytosol. Also because of what I told you in the slide of the coronavirus, the problem is that inside a cell and inside a virus, inside of any particle that is related to cell biology, the environment is extremely crowded, it's very densely packed. So this is a picture, or actually a schematic. It's not a real picture, but it's a schematic that tries to represent the concentration of molecules inside a living cell. So you see there's not a lot of space. That blue space that you could see in the previous slide is absent. The molecules are extremely densely packed, and this creates a lot of challenges for the cell. So the protein folding and the homeostasis of proteins inside a cell is a big challenge. So we try to understand, how does protein misfolding lead to disease, and how do cells respond to the accumulation of aggregated proteins when they misfold and accumulate inside the cell. We know, and I told you this before, that there is an age-associated decline in proteostasis. So proteostasis is protein homeostasis, and it's actually, we talk about the proteostasis network. So it's this network of different pathways in the cell that includes the ubiquitin proteasome system, the autophagy function, the molecular chaperones, and many other pathways in the cell that are related to the maintenance of protein homeostasis. And we know that with age, there is a decline in the function of some of these organelles, suggesting that because they may be failing with age, they are getting old and tired, maybe this is why then we start to have proteins misfold and accumulate and cause problems. That's just an hypothesis. In the context of Parkinson's disease, for those of you who are medical doctors, you will know this very well. It's known as a movement disorder because in the brains of patients with Parkinson's disease, there are some neurons in a region called substantia nigra that is shown here in these brain slices of the midbrain. So you see here a healthy brain, and you see this dark area here. These are neurons that contain a pigment called neuromelanin. And you see that in the brains of patients with Parkinson's disease, this area is not as dark. And this is because those neurons that used to be there, they died and they disappeared from the brain. And what we know is that usually when we look in those neurons in the substantia nigra, and then also in other neurons in other areas in the brain, But in these neurons, the ones that are surviving, we find these structures, these protein inclusions called Lewy bodies. And we know that these Lewy bodies seem to be composed of proteins, a variety of proteins, and some of these proteins can form these types of fibrils that you see here on the right side. And these fibrils are composed of a protein called alpha-synuclein. So alpha-synuclein is very important because it accumulates in Lewy bodies in the brains of patients with Parkinson's disease. But it's also important because mutations in this protein can be associated with certain monogenic but also idiopathic forms of Parkinson's disease. So it's implicated with the disease genetically and also neuropathologically. Now, we need to keep in mind something that often we tend to forget, which is that Parkinson's is much more than just a movement disorder. So this means that it's not just a disease of the substantia nigra, like we see here, but it's a disease that affects many other areas in the brain. And we don't know very well why this happens. We have some ideas at the moment in the field, but we don't know very well why those other brain areas are affected. And this is important because the way we treat Parkinson's will have to be not just about treating the problems that arise due to the problems in the substantia nigra, but also due to problems in other brain areas as well. So let's look at this protein, alpha-synuclein. Since it's such an important player in the disease, let's look a bit closer at this protein. And protein is represented here. So you can see amino acid, the N terminal of the protein, the C terminal. So the protein has 140 amino acids. We can define 3 main regions, 3 domains in the protein that are shown in different colors here. And then there's these different letters above the protein that I will come back to. We know that this protein is abundantly expressed in the brain. It seems to be presynaptic, so it exists in the synaptic terminals, but we also find it in other compartments. I will not explore this today, but we know that it's also present in the nucleus of cells, for example, and we don't know what it's doing there. So the function, when we talk about the function of this protein, we say that it's related to synaptic vesicle trafficking. The problem is that when we say that, we tend to ignore the fact that this protein is also highly abundant in the blood, in red blood cells. And we don't know what this protein is doing there. We just don't study this in great detail. And then we have several other pieces of information about what this protein looks like and how it is composed and what it might be doing. But the truth is we don't fully understand what this protein is doing. So when we come to these questions, why does this protein become toxic? How does it cause these diseases where it misfolds and accumulate that we call synucleinopathies? Then the question is, do we understand this? Do we have answers for these questions? And the truth is we don't. We don't understand how this protein goes from this status that we think is more the normal and functional state, where it has some alpha helical content and then some disordered regions as well. And how does it go from this to a Lewy body, to an insoluble protein aggregate that accumulates in the brain? And this is when we need to introduce another important idea, which is that proteins are much more than their primary amino acid sequence. So it's not just about knowing what are the amino acids that make the protein. It's about knowing what are the chemical modifications that these proteins undergo during their lifetime. And these we call post-translational modifications. So we know these are determinants of protein folding, localization, and function. And I don't know if there are any colleagues participating from Spain, from Barcelona. They would know this cathedral very well in Barcelona, but maybe a lot of you have traveled to Barcelona in Spain and have seen this. And the reason I'm showing this is because this cathedral has been under construction for more than 150 years. And this is in a way what happens to proteins. You know, proteins are being made in the cell, so you have the protein synthesized from the ribosome with the primary amino acid sequence. But then during the lifetime of the protein, there are different enzymes or processes that sometimes they don't even involve enzymes, as you will see later, that can add little chemical groups, so modifications to different parts of the protein. And then depending on the combination of modifications that the proteins undergo, they can lead to a conformational change, like the one shown here. So the protein changes its shape because the chemical groups that are added to the protein cause this change in conformation. And this can, of course, in the case of certain proteins like alpha-synuclein or other proteins related to other diseases, can change the way they fold, and this will change the way they function. And sometimes it even changes where they localize inside the cell. So these post-translational modifications, these are not happening there just to cause problems in proteins. They are actually tools that nature used to increase the complexity of proteome. So when we look at our genome, we know we have about 25,000 genes. Through alternative splicing mechanisms, we generate a larger number of transcripts, and then it's ultimately through post-translational modifications that we generate the complexity of the proteome that we need to have in each of our cells so that the cells can function normally. And so you can think of post-translational modifications as these condiments that can be added to proteins to change their folding and their function. So in a way to give flavors to proteins so that they can do whatever they need to do. And this is important because there's many proteins that we call intrinsically disordered proteins. So these are proteins that when we look at the structure of the protein, we cannot see a very well-defined structure. If we try to crystallize the protein or to use other techniques like nuclear magnetic resonance to study the structure of the protein, we cannot detect a very defined structure. So they're called intrinsically disordered. And these are not rare proteins. They are present in about 44% of the proteome. So about half of the proteome are proteins that don't have a very well-defined structure, which allows them to perform perhaps different functions inside the cell. And it's then through these post-translation modifications, these condiments, that we can adjust and fine-tune what these proteins do inside the cell. So we started to ask some questions such as, do specific post-translation modifications or combinations, sets of post-translation modifications, affect the aggregation of alpha-synuclein? And I'll show you some data related to one of these studies that we are conducting. And this is important because it connects Parkinson's disease with another major disease that I'm sure a lot of you deal with on a daily basis, or at least have heard about, because everyone knows about diabetes. So the question is, what is the relationship between diabetes and Parkinson's? And the reason I have this here is because we know that patients with diabetes have increased risk for developing Parkinson's disease, but we just don't know very well why. What are the molecular mechanisms that really connect these two diseases? So we think this is related to this type of chemical reaction called a glycation. And this is the non-enzymatic reaction of proteins with sugars. So if we have sugars in the cell, and we need sugars so that the cells can function, and if we have proteins with amino groups available, then a series of chemical reactions can take place. We can form a Schiff base, an amidori product, and then eventually there will be other products formed that may result in the formation of these advanced glycation end products, or AGEs. These are important because they are typically irreversible. It means that once these products form in proteins, they cannot be removed. And then the proteins, in a way, are damaged, and they may not behave and perform in the normal way that they should perform. So an important concept to keep in mind is that glycation is unavoidable. How can we avoid glycation? Well, we cannot, because all cells use sugars, or most cells use sugars to obtain their energy from. And this means that they will generate different products that are reactive. One of them, the most reactive product that is generated from glucose metabolism, for example, is methylglyoxal. Methylglyoxal is this aldehyde that is highly reactive and can react with different types of amino acids, such as lysines and arginines, and can form this type of advanced glycation end product that is shown here in this cartoon, that is called the carboxyethyl lysine, or CEL. And this is a product that forms when methylglyoxal reacts with lysine side chains in proteins. So you can think of your protein, as I showed you before, and you can think of a lysine residue here, for example, and if instead of being ubiquitinated, which is a tag that is normally used to label proteins for degradation by the proteasome, but if now this particular residue that would normally be ubiquitinated is blocked by glycation, then this means that the protein will not be normally seen and processed by the cell and will not be degraded and can accumulate and misfold and cause all the problems that we've talked about. So I'll show you here some data, some real data, where we looked at human brain tissue. So we got some human brain tissue from a brain bank, and we got samples from Parkinson's disease brains, patients with another synucleinopathy called Lewy body dementia, or control individuals, and we mixed up the samples. They were random, and then we used an antibody to detect alpha-synuclein. And you can see here in green the alpha-synuclein protein, which varies in levels. In some people there's higher levels, and some people there are lower levels of alpha-synuclein. And then we used an antibody that detects the carboxyethyl lysine modification. You see here in red that we can see some signal that overlaps with the signal for alpha-synuclein when we merge. Now, to study this in more detail, we went from the human brain to models, and we wanted to see and to try to understand what glycation does to proteins when we look in different model systems. So we used a variety of model systems. I will not show you all the data we generated, but we used very simple cells like the ones shown here, and these are, I'm sure a lot of you would recognize, these are yeast cells, and they are the same type of cells that we use to make bread and beer and wine, products that we all take in our daily lives. And we used also a variety of other model organisms, including Drosophila and mice and rats and primary neuronal cultures. So we think that by combining the use of different models, we may be able to get closer to understanding what is really happening. Because the truth is, all models are wrong, but some are useful. This is a famous quote by George Box. So this means that you may think, oh, but a yeast cell is very simple, so it will not tell us anything useful about Parkinson's disease. And this is not true. It just depends on the question we are asking. The truth is, even a mouse or even a non-human primate, a monkey, may not tell us everything we need to know about Parkinson's disease, because they are still not humans. And there are many things, many aspects of Parkinson's disease that we know that happen in patients that don't happen in all types of animal models. So then, using models, we used mass spectrometry. So we used mass spec to study the chemical composition of this protein alpha-synuclein. And what you see here is the mass spec spectra. And you don't need to pay a lot of attention to this, but basically the residues that are shown here in red are lysine residues that we found to be glycated. And we found that they primarily localize in the N-terminal region of alpha-synuclein. So all these Ks represent lysines. They are more in the N-terminal region of the protein, although there are other lysine residues in the protein shown in yellow, but those we didn't find to be glycated. So the ones that were glycated were the ones in red in the N-terminal region of the protein. And this does a lot of things to the protein. It changes its ability to bind membranes. I will not show you all the data. And the other thing it does is, for example, in patient-derived iPS cells, So these are cells that were derived from fibroblasts that were obtained from the skin of patients or control individuals. So here, fibroblasts from control individuals or from patients that carry a mutation that increases the copy number of alpha-synuclein. So these patients have a triplication of the gene that encodes for alpha-synuclein. This is why on a Western blot, when you compare the levels of alpha-synuclein in a control subject with the levels of synuclein in the patient that has a triplication of the gene, you see a lot more alpha-synuclein. So what we did also was to take the same cell from the patient and we knocked down alpha-synuclein back to normal levels. And you can see here the loading control. So these are equally loaded on the gel. And then we took those cells and we treated them with increasing concentrations of methyl glyoxal, this glycating agent that I told you about before. And you see the control line here in black. So this is how it responds to methyl glyoxal. And then when we treated the triplication line with methyl glyoxal, we saw that it suffered a lot more from the cytotoxicity than the control line. And if we knock down alpha-synuclein back to normal levels, then we reduce the toxicity of methyl glyoxal. So this suggested that even in patient-derived cells, if we treat the cells or expose the cells to concentrations of methyl glyoxal, the cells don't like it, and this seems to be related to the levels of alpha-synuclein that they produce. Then the next thing we did was to take mice, so either control mice, wild-type mice here on the left, or alpha-synuclein transgenic mice on the right. And we used stereotactical injection to inject methyl glyoxal into the substantia nigra of these animals on one side, and on the other side, we injected just PBS, so no methyl glyoxal. So you see the PBS injected animals here on the top, and the methyl glyoxal injected side here on the bottom. And in red, what you see are Th positive neurons, so neurons that produce dopamine, and you can compare the amount of red staining you have in the wild type animals and in the transgenic animals. So you see that comparing wild type with transgenic, there's not a big difference in terms of neurons. They have an equally high number of dopaminergic neurons. But if we treat the animals with methyl glyoxal, you see that in the wild type, you have still a large number of Th neurons, but in the transgenic, you lose a lot of the Th positive neurons in the substantia nigra, suggesting that, again, if you combine alpha-synuclein with methyl glyoxal, you have problems. So we did a lot of other studies to try to understand the mechanism. And what you see here is a study where we basically asked, okay, so we know that the Lewy bodies in the brains of patients are positive for alpha-synuclein, and they're also positive for ubiquitin. And we know that ubiquitin is important to target the protein for degradation by the proteasome. I already told you this before. So what happens when we stain cells? These are another type of human cells where we can induce the formation of these alpha-synuclein inclusions. So these spots you see here, they are alpha-synuclein inclusions. And we asked what happens in a control situation when we treat the cells with just a vehicle or when we treat the cells with methyl glyoxal. And what you see here is very striking. So we stain both cells for alpha-synuclein in green and ubiquitin in red. And what you see here is in the control situation, the spots look yellowish or orange. And this means that they are positive for both alpha-synuclein and ubiquitin. But strikingly, when we look in the cells treated with methyl glyoxal, we could see the alpha-synuclein spots. So the signal is there, but not the red signal. And this was very interesting because basically this is telling us that the protein is still aggregating, but it's not being ubiquitinated. So we are blocking the ubiquitin site as the protein is glycated, and this causes the protein to accumulate. So we are in a situation where we cause this proteostasis imbalance by glycation. So glycation makes the protein misfold and aggregate. And of course, we want to go to the other side. This is the bad side, right? Where we could treat the disease and treat the patients in the bedside. And so to try to do this, we used molecules that are scavengers of methyl glyoxal, so they react with methyl glyoxal, and these are just two molecules we knew from the literature. And what we did here is to treat flies. So I didn't show you all the data, but we had data in flies showing that methyl glyoxal also changes the behavior of the flies. And here, what we did was to treat these flies that express the alpha-synuclein protein with increasing concentrations of methyl glyoxal. And you can see here that with 100 or 300 micromolar of the molecules that are scavengers of methyl glyoxal, the movement performance of the flies improves compared to the PBS. So this is the vehicle control. So the flies that express alpha-synuclein, they don't move so well. And then when we feed them with food that contains these molecules, they improve. So if you compare just the green bars, you see that it gets better. And the higher the dose of the scavenger, the better the flies perform. So to summarize this part, I didn't show you all this data, but I showed you data in mice showing that methyl glyoxal together with alpha-synuclein causes problems. I didn't show you all the data in flies and in other types of cells, but I showed you data in the iPS cells that is confirming this effect. I didn't show you then a lot of details about what the glycation does in terms of the aggregation of the protein. But interestingly, what we found was that this type of glycation seemed to promote the formation of oligomers of alpha-synuclein and not necessarily fibrillar forms, which we think would be the more mature aggregates, but this is entering another topic. And I also didn't show you a lot of additional studies we did in simple model organisms like yeast and other cell culture models looking at toxicity and aggregation, but we did a lot of studies. And I showed you that methyl glyoxal can be scavenged by certain molecules and that this results in certain benefits in flies, so in vivo, in a living organism, and also in cell models. I didn't show you all that data. So what does this mean then in the end? Well, the idea here is not to say that glycation is the cause of Parkinson's disease, because we all eat sweets and cakes and we drink sodas. So in our food, in our diet, we have a lot of sugar and usually a lot of extra sugar compared to what we really need. So it's not that everyone that eats a lot of sugar will develop Parkinson's disease. The idea is more that if we have already other problems in our metabolism or in our cells, maybe glycation can be one of these agents that tips this proteostasis balance to the wrong side and promotes the misfolding and aggregation of proteins like alpha-synuclein, and this is why then it can be associated with disease. So I just want to finish with a very different type of idea so that you don't think that protein misfolding is always a bad thing. You know, I want to give you an example of a situation where we can use the knowledge that we are generating from studying this problem related to disease to make something that can actually be good. And so, you know, misfolding can be tasty. I would think that you probably prefer to eat a fried egg or a boiled egg instead of a raw egg, not just because it's tastier, but also because it avoids problems of infection with things you don't want to be infected with, of course, but it also becomes tastier. And so we've known for many years, for about 20,000 years, we started to domesticate animals and plants to make them work together with us and do things that we don't necessarily want to do. Poor animals, but sometimes they engineer the plants like corn to get the corn to be tastier or healthier, or in this case to have different colors. So we've been engineering animals and plants for many years, and now the idea is that we can also try to domesticate molecules and to engineer them so that they do things that are useful for what we want to do. So we can take advantage of billions of years of evolution, because evolution has done this naturally. So the reason why we are here talking today using the internet is because evolution has developed and adapted our proteins so that they give us the ability to be intelligent and to develop all these things that we use today in our daily lives. And if we look at the proteins that we can extract from the brains of patients with human diseases, they can look like this. You know, all these fibrils tangled up, and we know that this is probably not so good to have in the brain. But then we also know that in some organisms, like in yeast cells, there's a phenomenon called the prion phenomenon that tells us that a protein, the same protein, can exist in two different conformational states. One here in these cells on the left is soluble. So you express the protein, you fuse it with a GFP to tag the protein so you can see it under the microscope, and the protein is soluble, it's dispersed throughout the cell. But the same protein in other cells that have a different phenotype, the same protein can coalesce and form this bright fluorescent foci. So this is very strange. How does this protein exist in these two different states? Well, one possibility would be that the protein could be post-translationally modified. But in the end, what happens is that you can form similar types of fibrils using this protein from these yeast cells, and they have a very regular appearance. They look like nanotubes. They are 10 nanometers in width, and they can have several nanometers or hundreds of nanometers and maybe even micrometers in length. So when you look at these types of fibers, they suggest that perhaps if we are smart, maybe we can use them to make something useful. And that's what some colleagues have done with some of these amyloid fibrils. So they've studied them in detail to know how the proteins are organized in the fiber, and to try to use this process of self-assembly, because this happens naturally. You don't need to do anything. You just put the protein in a test tube, you shake the tube for some time, and then eventually the proteins start to change their normal folding. They form seeds, and then the seeds grow and the fibrils form. So this is what's shown here in this curve. So there's a lag phase when not much is happening in the tube, and then there's an elongation phase when the proteins are, the fibrils are growing, and then eventually you have the mature fibril assembled, where you have those properties that I showed you before. So the idea is that you can use some of the unique properties of these proteins, the fact that they assemble alone, so you don't have to do much to the protein and they form. They have useful properties for nanotechnology, for example, and you can then manipulate and produce larger amounts of these proteins. You can functionalize the proteins, adding different groups to give specific properties to the proteins. And I'll just show you, this is an old example now. It's a study from 2003, but it will just give you a general idea. Maybe with your creativity, you can do something even more creative in the future. So this is an electron microscopy picture. So the scale bar is here, 5 micrometers. You can see this is really small. And these are in black. You have 2 metal electrodes. And then these black dots, these are proteins that were added to these electrodes. And you see some amyloid fibrils here in black. Now, in this case, there's no fibrils connecting the two electrodes. So when you measure the current that is going from one electrode to the other, you cannot measure anything. There's no current going through. Now, in a situation where you can actually detect fibrils connecting the 2 electrodes, now all of a sudden you see that there's current going through. So you can use these proteins and these fibers to give them the properties that you need to have. So you can coat them with gold or any metal particle that will be a good conductor, and you can coat them with these metals, and now all of a sudden you can have tubes, these nanotubes that can be used for bioengineering, for example. So this is a situation where protein misfolding is not something bad. It can be used for something good. So that was just the final example I wanted to use. I want to show you here. This is an older picture of the lab. I need to have a new picture from this year, including one of the students that you already heard, Liana Shvachi. She's a PhD student working in my lab, so she should be here on this picture. I promise next time she will be there. And I want to thank a lot of colleagues that we've collaborated with over the years, funders for the support that they've given us for us to do the work that we're doing. And finally, for those of you who might be interested in learning more about alpha-synuclein, I leave you here a special issue that we edited last year, where a lot of colleagues that are experts in the field of alpha-synuclein have contributed to. So if you want to learn about this protein, I recommend you to search for this issue. You'll learn about a lot about different aspects of the protein, and hopefully you'll be interested in continuing and working even more in the future. So with this, I end, and I will be returning to the session here, and I'll be happy to take questions. I hope you could hear me well, and yeah, I'll be happy to take questions if there are questions, if there's a chance to ask questions now or later. Just tell me what to do.
Thank you, Professor Otiero, for an amazing lecture. Dear participants, please write your questions in the chat if you have any of them. We are waiting for your questions, please. Our participants are too shy. They are just writing something like very nice lecture, amazing lecture, but they do not write any.
Yeah, maybe it's difficult to come up with questions and to write them quickly here. But in any case, you know, I'll be happy to answer questions. You can also email me, and if you have any specific question, I'll be able to respond. And yeah, I don't want to take extra time. I know you have a tight schedule and you have a lot of other interesting lectures to listen to. So I wish you all the best for the meeting, for this new format. And again, it was a pleasure for me to be here and to contribute in this opening session. So I wish you all the best. And thank you again for the invitation and for the opportunity to be with you.
Thank you, dear Professor Otiero. We are thankful for being with us, even in this hard situation during the pandemic. And we really hope that after this all ends, we will be able to listen to your lecture, not only online. Thank you for your lecture, and thank you for everything.
Yeah, thank you, Anastasia, for all the work you've been doing. That's great. Thank you.
Amazing. So we have to continue our first plenary session. And now I would like to try to give a floor to our Mashkur Isa, who was not able to talk last time, but now I hope he fixed his microphone and he will be able to say what he plans to say.
And he will be able to say what he plans.
Hello, everyone. I'm sorry for the initial technical difficulties. I didn't realize my microphone was turned off. I'm Mashkur Isa, I'm actually part of the BIMCO family. I've been affiliated with BIMCO for several years now. And I'm proud to talk to you today in this new innovative form of BIMCO. Due to the coronavirus, there has to be a lot of changes, and people have to improvise, innovate, and find better means by which to continue their daily activities. This is one such way, and I'm happy that BIMCO wasn't cancelled, but it was instead transformed into an online session. I hope all of you are enjoying the lectures that are currently going on and will have a very fruitful experience. I'm a youth ambassador for the European Code Against Cancer. That's of the Association of European Cancer League. That's the ECL. So what is the ECL? The ECL is a non-profit pan-European umbrella organization of national and regional cancer leagues. Their major aim is to create a Europe free of cancer, and they're going to do this by influencing EU and pan-European policies. It's known generally that the cancer burden is going to increase by 35% in the year 2030. But this can be reduced by up to 50% simply by following the 12 recommendations of the European Code Against Cancer. The European Code Against Cancer are recommendations that were designed by experts aiming to reduce the burden of cancer. If you follow this code, the 12 recommendations include: don't smoke, that's reduce the tobacco use in any form, maintain a healthy body weight, be physically active in everyday life. Yeah, we know in the current situation of social distancing, everybody has to stay at home, and most people are actually sitting all through the day. But it's necessary for you to take action to maintain a healthy body weight, to be physically active. You should also have a healthy diet. If you drink alcohol, you should limit the amount of alcohol you use. You should avoid too much sunlight in your workplace. Protect yourself against cancer-causing substances. Reduce your radon levels. Women that are breastfeeding should breastfeed their children to reduce their cancer risk and also reduce the risk for the baby. And we should ensure that our children participate in vaccination programs, take part in organized cancer screening programs. All these recommendations are just aimed to reduce the cancer burden by at least 50%. If we follow these recommendations, we are sure to achieve the goal of a Europe free of cancer in the next few years. As a youth ambassador for the ECAC and also as a member of the organizing committee of BIMCO, we deemed it necessary because BIMCO follows the same ideals and the mission. They are passionate about seeing that Europe and the whole European regions are free of cancer. I'm happy to be part of BIMCO. The BIMCO family is amazing, and I'm sure you're all going to have a very fruitful and amazing experience. I wish you best of luck, and thank you very much.
Thank you, Dia Mashkur, for being our ambassador coordinator and for representing our possible partners, European Code Against Cancer. And actually, we had a few questions from our participants. Maybe they just didn't come in time, but Professor Otiero is ready to answer the question of our participant now after the talk of Mashkur is up.
Okay, so I'm back again to address a couple of questions, or one question in particular. So the question was whether there are any similarities between alpha-synuclein and tau proteins. So for those of you who may not know, tau is a protein that is associated with Alzheimer's disease and other diseases that are called tauopathies. So these are diseases where this protein tau misfolds and accumulates in the brains of patients with these diseases. And the idea or the question is whether there are similarities. And yes, the similarities exist in the sense that both proteins misfold and accumulate. We don't know also what tau is doing. We have some ideas. It associates with microtubuli in the cell, but we don't know all the possible functions of the protein. And we know also that there is an interaction between alpha-synuclein and tau that we don't fully understand. So there's many aspects that are similar about how these proteins misfold and accumulate in the brains. we are also thinking that it would be important to test whether glycation can also change and damage tau and cause it to misfold and aggregate. So it's an interesting question. There's many aspects that seem to connect tauopathies with synucleinopathies, and we don't fully understand how it works. So we are looking into this, and there's many groups in the world looking into this. Another question I see here is whether we tested glyoxal or other proteins glycated by glucose. And yes, so actually, this is another good question. So methyl glyoxal was the glycating agent I mentioned in our studies, because it's a very strong glycating agent. But even glucose can be a glycating agent, and even other sugars like fructose and galactose and ribose can be glycating agents. So what this means is that different sugars can modify proteins in different ways. And we know, because we are comparing all these sugars in parallel and the effects of these different sugars on alpha-synuclein, and we know that they induce different effects in alpha-synuclein, and they cause the protein to behave differently. So yeah, that's a very important question. Thank you for that. I think that's it.
Yeah, and unfortunately, I didn't see any more questions.
There's one more.
Yeah, sorry.
That I saw now, whether I can explain the physiotherapy of Parkinson's disease, whether it has advantage over other therapies. So physiotherapy, you all know what it is. It's about doing some specific exercises that will help your body function better. So there's different types of physical therapy or physiotherapy. And indeed, at the moment, since we don't have very good pharmacological therapies, physiotherapy is highly recommended for patients with Parkinson's disease. There are specific programs in the general societies that deal with Parkinson's disease. There are special programs of physiotherapy that Parkinson's patients can do to help them deal with their motor problems, with their movement problems. So these patients often fall, and they fall because they cannot control their walking, their gait, as well as people without the disease. And there are different physiotherapy strategies that help the patient. So this is something I definitely recommend. If you know someone with Parkinson's disease, please advise them to seek a specialized physiotherapist, because this can help them a lot. And I think that was the last question for now. So maybe.
Yes. Thank you very much, dear professor. Thank you one more time for your lecture and for all your answers, for the questions, and just for being our speaker this year. Thank you so much.
My pleasure. Thank you. Bye-bye.
Bye-bye. And now I have to inform all of the BIMCO participants that unfortunately our lecturer and true friend of BIMCO, Dr. Zulfikar Mirza from the United Kingdom, unfortunately will not be able to provide his lecture today due to the epidemiological situation and his personal participation in fighting with the coronavirus COVID-19 from health. And he really hopes that after the pandemic, he will be able to read not only one, but a lot of another interesting lectures. And now, before we start our separate meetings of the Congress, I would like to talk a bit the program of the Congress, just to remind you about meetings and events which are going to take place today and tomorrow. So separate meetings of the Congress will take place on the 7th of April from 1 p.m. to 7 p.m. and on the 8th of April from 11 a.m. to 5 p.m. Please download the program of the Bukovinian International Medical Congress 2020 and find your time of presentation and connect before your group starts its work. I would like also to remind about the second plenary session, which is a theoretical workshop, Study Smarter, Not Harder, Being Better at Medicine, from our partners AMBUS. It will start at 10 a.m. tomorrow on the 8th of April. Also, do not miss the third plenary session, and it is the lecture of our second speaker, Dr. Kamil Karimoglu, and he is going to talk about role of neuronal chromatin regulation in memory formation and cognitive disorders. It will start at 5 p.m. on the 8th of April, tomorrow. The closing ceremony of the Congress will take place tomorrow at 6 p.m. GMT +3. Do not forget to take some photos and tag us on our social media with our hashtags BIMCO 2020 and BIMCO Online. And now we have only half an hour before our separate meetings will start, so I recommend all of you to enter now your personal BIMCO account on our website registration BIMCO UA and follow the link of your section. You now have half an hour to have a coffee break and to get well prepared for your presentation. Do not miss your time, please. It is very important. And I wish one more time the fruitful work to every one of you. Let the Lord help you and see you during the separate meetings and the next plenary sessions. Thank you.
You are now currently the only person in this conference.